I'd like to thank the organizers for having invited me to this lecture. The title, as you can see it, is The Discovery of a Novel Family of Multifunctional Drugs, and we call it Serendipity in Full Force. For those of you who don't know what serendipity means, it's in Hebrew, mikriyut. You start out in one direction and you wind up in a different one. And instead of thanking my collaborators at the end of the story, I usually like to thank the people at the beginning of the story, and especially Shani and Sveta, who are here in the audience. The other collaborators from the Hebrew University didn't come. And this reminds me what Professor Danishevsky once said, that the word laboratory consists of two words, labor and oratory. They are the ones who did the labor, I will do the oratory. And a second thing, since we are waiting for the Minister of Health to come in, so I'll just tell you a little thing, is that this project began a few years ago when I gave a lecture about some anti-cancer agents, where we had shown that one of the compounds that we were working, the main compound that actually reached clinical trials, was able to cross the blood-brain barrier. And Professor Rosin, Marta Rosin, who was the recipient of the Pras Israel Prize a few years ago for developing a compound that we'll talk about it, approached me saying that she would like to see if the technology we had used for making the anti-cancer agent and cross the blood-brain barrier, whether we could do the same thing to help in the transport of her compounds. And the interesting part of the structure of this anti-cancer agent is that the structure is so simple that my daughter was able to knit a kippah with the structure on it. So this is the advantages of combining science and Judaism. And if nobody's here, I might, if he hasn't arrived, maybe I will continue, maybe we'll have to stop in the middle. Factors that contribute to Alzheimer's disease that are being studied and explored are the amyloid hypothesis with the accumulation of extracellular beta amyloid deposits, the cholinergic hypothesis that involves reduced synthesis of acetylcholine, oxidative stress, the presence of reactive oxygen species being superoxide radical, hydrogen peroxide, singlet oxygen, and free radicals such as nitric oxide, and inflammation. And as you can see, this is a very complicated disease. No wonder that we are at a loss after having spent, the world has spent billions upon billions upon billions of dollars trying to find a cure for this disastrous disease. We are still on the first stage. There is nothing that we can show for it. Speaking about the cholinergic effect of the involved in the drug, where people who suffer from Alzheimer's disease display a diminished production or diminished presence of acetylcholine in their brains. And acetylcholine, I just schematically show here, undergoes catalyzed by acetylcholinesterase hydrolysis to give choline and acetic acid. And people who suffer from a lack of acetylcholine, the presence of acetylcholine, we try to inhibit the activity of the acetylcholinesterase. And they have been found compounds natural compounds and other synthetic ones that inhibit the enzyme, and these compounds are usually carbamates of aromatic systems, such as neostigmine, that by doing a carbamylation of the serine group, temporarily inhibit the enzyme, and therefore, if the enzyme is inhibited temporarily, it appears that the small amount of acetylcholine that is being produced is in a larger concentration. And indeed, a number of compounds have reached the market for the treatment of the early stages of Alzheimer's disease, of which all of them are acetylcholinesterase inhibitors, of which the compound called Rivastigmine is the one that Professor Rosin developed. And let's put it this way, it is considered the most popular compound for the treatment of this thing. If you can say, it's very successful because it sells for about $1 billion a year. So it's a very successful drug. Whether it actually does much is a questionable situation, because it maybe helps in the early stages of the disease, but certainly does not solve the problem. And one of the questions that we asked ourselves is that whenever you have these highly complicated diseases, cancer, schizophrenia, Alzheimer, that are multifactorial diseases, it is almost impossible to imagine that you will be able to treat this drug with a single agent. Usually what you will do, you will take a whole bunch of compounds. And what we asked ourselves, the question is, could we design a molecule that is active at least against 2 or more of the factors that contribute to the disease? And not a long time ago, I came across this very interesting article by Michaela Rossini, that she speaks about polypharmacology, the rise of multi-target drugs that happen to have interesting properties, because instead of dealing with 2 different drugs, each of which has different pharmacokinetics, different interactions between one and another, if you get everything into one single drug, you may be able to show some advantage. And since our distinguished minister has arrived, I will take a break and I will give him we'll try to continue. The compound on top on the left is the Rivastigmine, the compound that Professor Rosin developed as a cholinesterase inhibitor, based primarily, as we can see, on the presence of the carbamate functionality attached to the aromatic ring. A second compound that she has been developing over the years is this compound called ladostigil, which also has a similar type of functionality. Now, please note from the structural point of view that in this case, the nitrogen atom is separated from the aromatic ring by 1 carbon, whereas in this case, the nitrogen atom is attached directly to the aromatic ring. In our bodies, we can find 2 other compounds that are very well known and well established. One of them is melatonin, that is involved in the circadian cycle of the sleep and awakeness, and the other one is indole acid. These compounds, as you can see from the reported articles, melatonin displays radical scavenging properties. It's also presents some anti-inflammatory effects. Indole propionic acid shows oxidative stress. So if we look right now at the combination of these compounds, we see that we can have a commonality in the structure. All of them have this aromatic ring, where we have the nitrogen in close proximity to the aromatic ring. And being that these display, as I say, cholinergic activity, and these display antioxidant and oxidative and anti-inflammatory properties, we decided to explore a project based on the following. Let's build molecules that will have the structural features of all of these together, namely the indolic species that has here either an acidic or a basic group, plus the carbamide function, and see if this particular type of compounds will display everything that we said, namely cholinergic activity, as well as antioxidant and anti-inflammatory activity. And indeed, we began making these compounds, and we made a lot of them. By the way, in the course of this project, we have already made more than 500 compounds, including the intermediates, and this is for a laboratory in which we have just a few people working. So, with a little bit of effort and good luck of having good students, you can produce a lot of chemistry. The problem became evident quite early in the game, because these indols are very insoluble in water. And therefore, even for the early evaluation, it became difficult to administer these things. So we figured, let's see what will happen if we will reduce the indole to the indolic system, indolinic system, from an indole to an indolin, where the nitrogen becomes basic, and therefore we should be able to make salts of these compounds, and they will be water soluble. Indeed, it worked. All the compounds that we made were very water soluble, and the first case of our serendipity was that these indolinic compounds were much more active than the corresponding indoles. However, one problem that appeared is that upon reduction of this double bond, we are creating on position 3 of the system in a chiral center. And as you well know, always chirality becomes a little bit problem. You have to separate the enantiomers, etc., and it just makes life a little bit more difficult. So we figured, let's see what will happen if instead of attaching the chain to the position number 3, what will happen if we attach it to position number 1? If we will do this, the chirality of the molecule will disappear, and therefore we may get some interesting compounds. Indeed, when you overlap the structure of these two compounds, you can see that they are virtually identical. So we switched at this point to concentrate our efforts to these indolinic compounds. And as you will see here in several diagrams, we prepared a very large number of them. Indeed, initially, all of these had the carbamide function, but they also display the antioxidant and anti-inflammatory properties that we were looking for. But not only this, these other properties, the antioxidant and the anti-inflammatory properties, were achieved at extremely low concentrations, much less than the concentration of the compound needed to display the cholinergic activity. And moreover, at this point, somebody pointed out to us that excessive cholinergic activity can cause all kinds of other problems. And being that the chances of finding something for the treatment of Alzheimer's is rather remote, we switched our program completely away from Alzheimer's, and we began to investigate the antioxidative and anti-inflammatory properties of the compounds. And in one example, just to show you, we began with this benzyl-protected indole, which we condense with acrylonitrile and then reduce the indole. We reduce the indole to the indolin and reduce the nitrile to the amine, and the next step was going to be a debenzylation, a catalytic hydrogenation for the removal of the benzyl group. So that seemed to be a rather straightforward experiment. However, to our great surprise, instead of getting what we wanted, namely this compound, we found out that, and for all the students pay attention, the PAR apparatus in which we were running the hydrogenation had been cleaned before with some acetone, and traces of acetone remained in the system. And what happened is that the intermediate that we made reacted with the acetone, and upon reduction, it, instead of giving us the primary amine, gave us the isopropyl amino derivatives. And serendipity working full time, we found out that these isopropyl amino derivatives appear to be the most active compounds we ever encountered, as you will see later. So don't despair if your students make an error by night cleaning everything, but serendipity works. So here we continue making the synthesis. Just for one curious synthetic problem is that in some of these compounds, we had to carry out 3 or 4 different types of hydrogen reduction reactions. And we had to play around with these to find out how do you reduce the indole, how do you reduce the double bond on the side chain, how do you reduce the nitrile. All these things, we were able to find the right conditions to selectively reduce every single one of these double bonds, and indeed the number of compounds produced were tremendous. All this work was continuously being carried out in collaboration with Professor Rosin and her people, where we would send the compounds to Jerusalem, and there they would be tested. And I will go right now, we'll leave the chemistry. Let's see some of the biological data that was generated. The first thing, we're going to talk about the oxidative stress. As I mentioned before, among the reactive oxygen species are also nitric oxide is one of the natural insults, and therefore we try to determine whether our compounds would reduce the nitric oxide. And as you can see, our compounds, which are listed here, are just as active or much better protectors of nitric oxide damage, which is induced by administering polyliposaccharides to the cells. This causes this leads to the secretion or the formation of the nitric oxide, and therefore we will try to reduce it. And indeed, they are just as good as this budesonide, which is a steroid. A second protective effect against oxidation is the survival of the cells against injury caused by hydrogen peroxide. And in this case, we wanted to compare it to plain indolin, without any substituents, just to see if it could have been that the activity that we were observing was from just such a simple compound that is well known from many, many years, but had never been tested for any biological activities. And indeed, the compounds, all of them showed this potent anti-oxidative effects, much better than the Indolin. So these were two in vitro experiments that just showed the antioxidant effects. Here is some of the mechanism. I don't want to get involved in this. If anybody is interested, we can discuss it. And then we wanted to evaluate the anti-inflammatory effects. Now, chronic activation, I'm going to read this for you. Chronic activation of the immune system and the release of pro-inflammatory cytokines, TNF-alpha and interleukins, occurs in a variety of disorders, including ulcerative colitis, Crohn's disease, rheumatoid arthritis, diabetes, chronic liver disease, atherosclerosis, multiple sclerosis, and many other diseases. And this is, again, one of the reasons why we switched to the anti-inflammatory effects of the compounds, because there are so many diseases where the inflammation is a major contributor to the disease that we would have a much better chance of finding some desirable compounds that will be useful for any of these diseases. Frequently, chronic inflammation and oxidative stress are major contributors to a large variety of diseases. As you can see here, Alzheimer's, pulmonary arthritis, diabetes, cancer, etc. And drugs that inhibit the excessive release of these cytokines, the TNF-alpha and the interleukins, may be of therapeutic benefit for these diseases. I'll give you here a few examples right now of some of the activities that our compounds show in actual diseases. One of them is the acute liver failure. This disease relates to the loss of liver function that occurs rapidly, sometimes in days or weeks, usually in people who have no pre-existing disease. The acute liver failure is less common than chronic liver failure, which develops more slowly. Acute liver failure is a severe, life-threatening clinical syndrome caused by the sudden and severe liver injury. The underlying pathological cause for this disease is extensive necrosis and apoptosis of the hepatocytes induced by acute bacterial viral infections, excessive alcohol intake, drug overdose, all kinds of causes for this thing. And we can cause in the animals a synthetic acute liver failure by administering to the animals galactosamine. Here is the structure of this sugar, and in combination with these lipopolysaccharides, this combination induces an acute liver failure through the elevation of these TNF-alpha and IL-6 cytokines. They cause the damage resembling that observed in the human disease. The current study, we evaluated 2 of our compounds. And you can see here in this picture, this is the control liver tissue. This is the liver tissue treated with the steroid dexamethasone. This is just the damaged liver by being treated with the galactosamine and the lipopolysaccharides. And this is the protective effect that 2 of our compounds are given to this thing, a much diminished damage to the liver by this treatment. But moreover, pay attention that these compounds are being active at these very, very small concentrations, whereas dexamethasone, which is a steroid and has a lot of side effects, is to achieve the protective effect, you need it at a much, much higher concentration. By the way, all the compounds that we have tested, we have not detected any toxicity. Here is the survival effect. As you can see here, when just the week after you administer the galactosamine and the polyliposaccharides, the animals die very rapidly, and within 96 hours, you have killed 90% of the animals, whereas the compound 1297 gave about a 50% protection, whereas 1284, the compound that had the isopropyl amino group that I told you before, protected about 80% of the animals. Another, let's call it general disease that we studied was what we called endotoxemia. Endotoxemia is a condition that can lead to septic shock, septicemia, by the addition of the lipopolysaccharides. And this is in the derivatives, our compounds reduce in the plasma the concentration of TNF-alpha and IL-6 to, for instance, the IL-6 can be reduced essentially as much as the dexamethasone does here. Not only that, it can also protect specifically the spleen, the liver, the brain, whereas in all of these cases, we have severely reduced the elevation of the cytokines. Another disease that we have explored is the Crohn's disease, and it is possible. Crohn's disease is a general inflammation of the whole digestive system. Colitis is primarily of the colon, and we can induce this Crohn's disease or an analogous Crohn's disease in rats by treating them with dinitrobenzene sulfonic acid at the concentration of 35 milligrams per kilogram. The effects of this compound resembles Crohn's disease in humans with the appearance of all these different factors. The inflammation begins within 3-4 days, and we administered our drug. Professor Rosin found a very good method of administering rectally, and the results were, as you can see here. This is the tissue, the colon tissue, the colonic tissue in the control where there is no disease. Upon addition of the dinitrobenzene sulfonic acid, you can see essentially almost total destruction of the tissues of the colon, whereas upon addition of one of the early compounds that still had the carbamate, we can see essentially full protection and retention of the structure of the colon. Now, we have examined not only these, we are collaborating with a large number of different groups, each one of which has expertise in different diseases. Many of these diseases are problematic because we have to cause the disease in the animal. And the Helsinki committees don't allow us to do these things. They say it causes too much damage, too much pain, too much discomfort to the animals. We have found, for instance, that in diabetes, we can take animals that are already have the developed the precondition of diabetes. And when we treat this, we see major benefits of these things. I couldn't give you so many of these things. By the way, I don't understand many of these things because all these are a variety of biological tests being carried out in a huge number of inflammatory diseases. And to our great delight and surprise, our compounds seem to act against many of these things, but not the same level. Some of them are much more selective for this disease or for that disease, which in turn implies that maybe there are different receptors in each and every one of these cases. So the data seems very promising, and my son happens to be, who works in Teva, happens to be in a meeting right now in Prague, where he met representatives of many of the companies, and he already contacted two of the major pharmaceutical companies, well known, I don't want to mention names yet, that appear to have some interest in this. We are hoping to be able to come to some of these companies and discuss our compounds, which we have patented them extensively. Some of the references that of the articles and patents that we have generated appear here. And just to finish the lecture, I'd just like to quote from Lednicer books. Has it ever occurred to you that medicinal chemists are just like compulsive gamblers? The next compound will be the real winner. And the second thing was the three princes of Serendip, Balakrama, Vijayo, and Rajashia, whatever his name was, as they traveled, were always making discoveries by accident and sagacity of things they were not in the quest of. So this is the story of the present project that we have, and I invite you to go to use a serendipity generator. Thank you very much.
Thank you. Thank you very much.